Bismillahirrahmanirrahim. Assalamualaikum and good day to all of you. I am Dr. Ani. I will continue the lecture for topic 11 on protist evolution and diversity. In Meta textbooks 13th edition, this topic are referred to chapter 21, page number 373 until 393. In your lecture note, we have outlined 5 subtopics for topic 11. Begin with the general biology of protists, followed by supergroup Archaeoplastida, and then supergroup SAR, then supergroup Excavata, followed by supergroup Amoebozoa and Opisthoconta. In this video lecture, I will explain all in once, so I will not divide this video lecture into several subtopics. By the end of this video lecture, you will be able to differentiate each supergroup of protists, and then you can relate protist evolution based on the characteristic for each supergroup. Let's see the brief explanation about kingdom Protista.
Of all the kingdoms of life, Kingdom Protista is perhaps one of the most diverse and interesting. Kingdom Protista spans from single-celled organisms with various means of locomotion and obtaining energy to huge kelp, hundreds of feet in length, that create expansive underwater forests that are home to a wide variety of marine life. But what do these diverse forms of life have in common? First, the cells of all protists have a nucleus and other complex organelles, unlike the prokaryotes of the domains archaea and bacteria, which have neither nuclei or complex organelles. Secondly, all the members of Kingdom Protista have one or more characteristics that prevent them from being classified in kingdoms fungi, animalia, or plantae. For example, though there are protists that bear a striking resemblance to fungi, molecular analysis reveals that the cell walls of these protists are made up of cellulose, not the chitin found in all fungi. Another example is that, though a form of green algae now classified in Kingdom Protista is believed to have given rise to the members of Kingdom Plantae, the method of reproduction nevertheless differs significantly between algae and plants. Finally, though there are animal-like protists which are believed to have given rise to all the larger organisms found in Kingdom Animalia, these single-celled protists aren't classified as animals simply because, by definition, animals are multicellular.
Protists are such a diverse group because it is a category many organisms are put into when they don't quite meet the requirement for a plant, animal, or fungi. Most are single cells, but some exist as colonies of cells or are multicellular. Protists are sometimes grouped according to how they acquire organic nutrients. They can be autotrophic, heterotrophic, or mixotrophic. Autotrophic is where the protists obtain the organic compound from photosynthesis process. Heterotrophic, the protists obtain the organic compound from the surrounding environment. Meanwhile, mesotrophic protists, they are able to combine both autotrophic and heterotrophic nutritional modes. For example, in this figure, we have Euglena viridis. Euglena viridis can obtain energy from a variety of sources depending on the environmental condition. They can photosynthesize in light and also hunt in the dark. In terms of protist life cycle, most protists reproduce asexually by binary fission or mitosis, where they produce identical daughter cells. But some protists also reproduce sexually, where they can produce genetically different offspring. It happens when there is an unfavorable condition where the crowded environment or food shortage. They will form spores and survive in hostile environment. This video shows the binary fission process by Euglena. Okay, now let's see the evolution of protists. Because of the complexity and diversity of protists, it makes them difficult to classify. From the DNA evidence, protists are not monophyletic, meaning that they do not belong to the same evolutionary lineage. As you can see from figure, there are three common ancestors of protists. Euglenozoa, Straminophylla, and Alveolata is evolved from one ancestor. Meanwhile, Rhodophyta and Chlorophyta evolved from another ancestor, and Coenoflagellida also evolved from another ancestor. Protists, land plant, fungi, and animal are now classified into 5 supergroups. Supergroup is high-level taxonomic group, below domain and above kingdom. There are Archaeoplastida, Sar, Excavata, Amoebozoa, and Opisthokonta. This figure summarizes of all supergroups under Protista. Archaeoplastida, Sar that contain subgroup Straminopyle, alveolates, and Rhizaria. Next is Excavata, Amoebozoa, and Opisthokonta. Okay, now we discuss about the supergroup Archaeoplastida. Archaeoplastida include land plants and other photosynthetic organisms, such as green and red algae, that have plastids derived from endosymbiotic cyanobacteria. In this supergroup, we will discuss about the evolutionary history and the example of the members. For example, green algae and red algae. The endosymbiotic theory proposes that eukaryotic cells acquire mitochondria and plastids, including chloroplasts, by engulfing a free-living bacterium that develops a symbiotic relationship within the host cells, a process termed endosymbiosis. Mitochondria were derived first from the endosymbiosis of an aerobic bacterium, and then chloroplasts were derived later from the endosymbiosis of a cyanobacterium. This will evolve becoming a red algae and green algae. The unique feature of Archaeoplastida, they have double membrane plastid. Plastid is an organelle to make foods that contain pigment. The green algae are protists that contain both chlorophyll A and B. They inhabit a variety of environments including oceans, freshwater, snowbanks, the barks of trees, and the back of turtle. Green algae can be found in numerous forms, for example in a single cell such as Chlamydomonas, in a colony such as volvox, and in multicellular forms such as urba. Plants are considered to be most closely related to the green algae because of plant and green algae have both chlorophyll A and B, a cell wall that contains cellulose, and food reserve made of starch. Molecular data suggests that green algae are subdivided into two groups, the chlorophytes and the charophytes. The land plant are thought to be derived from charophytes. There are two types of green algae. First, chlorophytes or chlorophyta, and second is charophytes or charophyta. For chlorophytes, first example is Chlamydomonas. Chlamydomonas is a minute unicellular green algae which is less than 25 micrometer long. It has a cell wall and big cup-shaped chloroplast with a pyrenid for starch synthesis. There are two long whip-like flagella project from the interior end to propel the cell toward light. Next example for chlorophytes is volvox. Volvox is a colonial green algae. A colony is a loose association of independent cells. Volvox is a hollow sphere with thousands of cells arranged in a single layer. Volvox cells cooperate when flagella beat in a coordinated fashion. Some cells are specialized for reproduction, and each of these can divide asexually to form a new daughter colony within the parental colony. Daughter colonies are inside a parent colony until an enzyme dissolves part of wall so that they can escape. Next example for chlorophytes is ulva. The multicellular ulva is called lettuce because of its leafy appearance. The body of ulva is 2 cells thick and can be as much as a meter long. Okay, now we see the example for charophytes. Charophytes are filamentous algae. The filamentous are end-to-end change of cells. Charophytes have both branched and unbranched filament. For example, Spirogyra is an unbranched charophyte, can be found on the surface of ponds and streams. Spirogyra, it has a ribbon-like spiral chloroplast. The other example for filamentous alga is Chara. Chara consists of stem-like body with nodes and internodes. Cells of the body originated from apical meristem that are homologous to plants. DNA sequencing data suggests that among green algae, the Chara or the stoneworts are most closely related to land plants. The other members in supergroup Archaeoplastida is red algae. Red algae is multicellular algae that live in warmer seawater. They possess red and blue pigments in addition to chlorophylls. They can be found in the form of filamentous with branch, feathery, flat, and ribbon-like form. Coralline algae are red algae with cell wall that contain calcium carbonate which contribute to coral reef. Red algae are economically important. They can be used in the making of agar. Agar is a gelatin-like product made primarily from the algae Gelidium. It is the source of agar used in the drugs capsule, dental impression, cosmetics, culture medium, electrophoresis, and food preparation. Carrageenan from red algae is an emulsifying agent used in the production of chocolates and cosmetics. The black wrapping around sushi rolls consists of processed porphyra blades. The second supergroup under kingdom Protista is SAR. SAR stands for Straminophiles, alveolates, and Rhizaria. These three is subgroup under SAR supergroup. The unique feature for this supergroup, they contain plastids more than 2 membranes. This figure shows the formation of alga groups under SAR by endosymbiosis process. Okay, for first or primary endosymbiosis, the organism will get 2 membrane plastids or organelle. If they undergo secondary endosymbiosis, they will get more than 2 membrane, the organelle formed inside the organism. The members under subgroup Straminophiles are brown algae, diatom, golden brown algae, and water moles. The members of Straminophiles have unique characteristics where they have two unequal flagella, hairy and hairless flagella. The members include brown algae, diatoms, golden brown algae, and water mole. The brown algae have chlorophyll A and C and accessory carotenoid pigment that give them their color. Their reserve food is a carbohydrate called laminarin. Laminaria, commonly called cap and fucus, known as rockweed, are examples of brown algae that grow along the shoreline. They cling to rocks with a structure called holophast. The example of brown algae is Laminaria, fucus, microcystis, and neurocystis. Brown algae are harvested for human food and for fertilizers. Macrocystis is the source of algin which is added to the product to give them a stable, smooth consistency. Diatoms are single-cell straminopiles that have cell walls consisting of two silica-impregnated halves or valves. Diatoms have a carotenoid accessory pigment which gives them an orange-yellow color. Diatoms make up a significant part of plankton, which serve as a source of oxygen and food for heterotrophs in both freshwater and marine ecosystems. Next example of traminophiles is golden brown algae. Golden brown algae have distinctive color. They derive their color from yellow to brown carotenoid accessory pigments. They are unicellular or colonial protists that typically have two flagella with tubular hairs. Many of them are mesotroph. For example, in this figure, Acromonas verrucosa, they can photosynthesize and phagocytize at the same time. Golden brown algae contribute to freshwater and marine phytoplankton. Next is water moles. Water moles are fungus-like protists, often seen as furry growth on their food source. Some live on lands and parasitize insects and plants. Water moles have filamentous bodies similar to the hyphae of fungi, but their cell walls are composed of cellulose instead of chitin. The second subgroup or supergroup SAR is alveolates. Alveolates have unique characteristics where they have a small air sac known as alveoli just beneath the plasma membrane, and all of alveolates are single cells. The member under alveolates is dinoflagellates, ciliates, and apicomplex. Dinoflagellates are single-cell photoautotrophic algae encased by protective cellulose and silica plates. Most of them have two flagella. One lies in a longitudinal groove and acts as a rudder. The other one is located within a transverse groove, and its beating causes the cell to spin as it moves forward. Some species, such as Noctiluca, are capable of bioluminescence. Zooxanthellae are dinoflagellates that form symbiotic relationships with invertebrates such as corals. The zooxanthellae provide their animal hosts with organic nutrients, and in return they receive shelter, nutrient, and protection. Under unusually high nutrient conditions, population of dinoflagellates and other algae can undergo a population explosion called alga bloom. For example, Alexandrium catanella in this figure caused red tide that caused massive fish kills by producing harmful neurotoxin. Another example of alveolates is ciliates. Ciliates are among most complex protozoan. There are single-cell protists moved by means of cilia, and they ingest their food by sweeping down a gullet and eliminate waste through anal pore. Most cilia have two different nuclei. The macronucleus controls the normal metabolism of the cell, and micronucleus is involved in sexual reproduction. The example of ciliates in this figure is Chilodonella and Paramecium. Next member in alveolates is apicomplexans. Apicomplexans, also known as sporozoans, are non-motile, parasitic, and spore-forming protozoan. All apicomplexans are parasites of animals, and they have unique organelle called apicoplast, which is used to penetrate the host cell. The most serious parasitic disease of humans is malaria, infected by Plasmodium species. Plasmodium is a parasitic protozoan that infects human red blood cells. Chills and fever appear as red blood cells burst and release toxins into the blood. The transmission cycle of Plasmodium involves the Anopheles mosquito as a vector that transmits the disease between the host and the other organisms. The last subgroup for SAR supergroup is Rhizaria. Rhizarians consist of foraminiferans and the radiolarians. Foraminiferans and radiolarians have a skeleton called test made of calcium carbonate. Foraminiferans often have a multi-chambered test. Its pseudopods extend through holes in the test. In the radiolarians, the glassy silicon test is internal and usually has a radial arrangement of spine. The pseudopods are external to the test. Tests of dead foraminiferans and radiolarians form deep layers of ocean floor sediment. Their fossil date as far back as the Precambrian, and they are evidence of the antiquity of the Protist. The third supergroup of Protista is Excavata. Excavata have unique feature with a typical or absent mitochondria and distinctive flagella and deep oral groove. The member under Excavata is euglenids, parabasilids, and diplomonads, also kinetoplastids. Euglenids is small freshwater unicellular organism. There are very diverse group and that's why it's difficult to classify. Euglenids contain chloroplasts surrounded by three membranes instead of normal two membrane. The special region of the chloroplast called pyrenoids. Pyrenoids produce unusual type of carbohydrates called paramylon. Euglenids have two flagellas with unequal length and eye spot for photoreceptor. The cell is bound by flexible pellicle that contain protein bands side by side. The example of euglenids is euglena. Next is parabasilids. Parabasilids are single-cell flagellated protozoan that are endosymbionts of animals and can survive in low-oxygen environment. Parabasilids have a fibrous connection between the Golgi apparatus and flagella. These protozoa lack mitochondria. They rely on fermentation for the production of ATP. Example for parabasilids is Trichomonas vaginalis. Trichomonas vaginalis is a sexually transmitted organism that causes vaginitis in females. Infected males may have no symptoms. Diplomonas is unicellular organism. Diplo meaning they have two nuclei and two sets of flagella. They are well known for causing various diseases in humans. Example for diplomonas is Giardia lamblia. Giardia lamblia is the most common flagellated in the human digestive tract. Diseases of Giardia lamblia are transmitted in contaminated water and cause severe diarrhea. The last member of supergroup Excavata is kinetoplastid. Kinetoplastids are single-cell, flagellated protozoan named for their kinetoplasts, which are large masses of DNA found in their mitochondria. They are well known for causing various diseases in humans. For example, Trypanosoma brucei are parasitic kinetoplastids transmitted by the bite of tsetse fly and are the cause of African sleeping sickness. Another example is Trypanosoma cruzi that cause Chagas disease in humans. In this figure, it shows the Trypanosoma cruzi with the triatomin bugs, the vector for the Trypanosoma cruzi, and right figure is Trypanosoma brucei. The second last supergroup under kingdom Protista is Amoebozoa. Amoebozoa have unique feature where they can move by pseudopods. The member under Amoebozoa are amoeboids and slime molds. Amoeboids are protists that move and ingest their food with pseudopods. Amoeboids feed by phagocytosis, by which pseudopods surround and engulf the prey. For example, Entamoeba histolytica is an amoebic parasite that invades the human intestinal lining. Slime mold are important decomposers that feed on dead plant material. They were once classified as fungi, but unlike fungi, they lack of cell wall and they have flagellated cells at some time during their life cycle. The last supergroup under kingdom Protista is Opisthoconta. Unique feature for Opisthoconta, they have flagella. Some of them have flagella and there are unicellular and multicellular. Animals and fungi are opisthocoans as are several unicellular and multicellular protists. The choanoflagellates are animal-like protozoan that are closely related to sponges. They include unicellular and colonial forms and are filter feeders. Most feed on algae or cyanobacteria. Okay, we have discussed all the 5 supergroups under kingdom Protista that include Archaeoplastida, Sar, Excavata, Amoebozoa, and Opisthoconta. Under Sar supergroup, we have discussed about the subgroup of Straminopiles, Alveolates, and Rhizaria. The most important in this topic is you can differentiate each supergroup based on their unique feature, and please remember the members of each supergroup. So that's all. Hope you understand, and the most important is you can remember each of these protista.